Welcome to the video for chapter 12.5. We are going to be talking about the influenza virus, which is an RNA virus. This is a pretty timely one. We're getting ready to head into flu season, right? We've all had the flu at some point in our lives. So this one's a little bit interesting because it's going to help you understand why the flu vaccine doesn't work some years and why it does other years. So let's get started. Okay, before we get into talking about the influenza virus, let's do a quick review question. New HPV virions are released from the body by A. Exocytosis, B. Endocytosis, C. Becoming enveloped in the cell membrane, or D. Shedding of host cells. So take a moment, pause the video, and see if you can't find the answer. Okay, so the answer to this one is D. The shedding of host cells. This is what happens when that basal cell layer becomes keratinocytes, right? They're able to be shed from the host cells. Okay, so the answer is D. Okay, before we dive into 12.5, let's talk about the section objectives. One of the things you're going to be able to do is describe the structure of the influenza virion. You'll be able to explain how the influenza genome enables rapid evolution of novel strains that cause pandemics. You'll be able to describe the replication cycle of the influenza virus. Okay, before we get into the nitty-gritty of the influenza virus, let's talk about some flu stats. Everyone watching this video has probably had the flu at some point in your life. About 10% of the US population gets at least one strain of the flu every year. We have roughly around 36,000 deaths on average each year. Most of these deaths occur in the elderly population of 65 years or older. Now, if we look at 2023 specifically, since that's the last year we have statistics, well, there was about 28,000 deaths from the influenza virus. Now, this is what we think is a lot, but let's do some comparisons, right? We just got done with a pandemic. So in the early years of the COVID-19 pandemic, in 2020, we had 352,000 deaths from COVID-19. We had more than 460,000 in 2021. Thankfully, after 2021, with the advent of the vaccine, we saw the number of deaths from COVID-19 decline to about 246,000, and they've declined even more since 2022. So there was a 70% decrease from 2022 to 2023 in deaths from COVID-19, where they have recorded around 76,000 in 2023. Now, what we're seeing here is that COVID-19 is becoming more and more like the flu virus the longer it is around. So one of the things that is characteristic of the flu virus is that it has high amounts of antigenic shift, not drift, shift. Okay, antigenic shift is a lot like antigenic drift, except it's bigger changes. Okay, it leads to more quickly having the virus evolve. Okay, and it's the main reason why we have to have a new vaccine every year for the flu virus, because of this antigenic shift. Okay, this antigenic shift is due to the structure of the viral genome in influenza. It is composed of viral genetic segments, which are sort of like chromosomes, but not. Let's look at this in a little bit more detail. So what do I mean when I say that the influenza virus is segmented? So the influenza virus has a segmented genome, which means it's in pieces. Now, this is very interesting because this is what's going to allow it to go through antigenic shift. Okay, those different segments can reassort themselves and cause a new strain to develop. Now, with influenza, we know that it's not restricted to just humans, right? We have the bird flu, we have the swine flu, we have the human flu. So let's pretend we are a farmer. We have a farm. Okay, maybe we have some ducks or chickens on this farm. Okay, these ducks and chickens can be infected with the bird flu virus. Okay, we could have the human virus. And let's say that one of our pigs doesn't have any virus at all. He's healthy. Okay, so what can happen is, because it can jump species very quickly, our pig can pick up the virus from the duck that he interacted with, and the human flu virus from when you fed him. Okay, so now he has both of these strains of virus inside. What's going to happen is, because it's segmented and it's in these little pieces, what can happen as these viruses are replicating, when they're repackaging themselves, we can take a few pieces from the bird flu and a few pieces from the human virus, and we can make a completely new virus. This is what is called antigenic shift. And this is why we have so many different strains of the flu virus every year. Like, you could get one strain of the flu virus and get a completely different one because of antigenic shift throughout the flu season. Okay, so now we're going to have segments from both viruses that are going to make something new. This is why we need a new vaccine. This is why sometimes the vaccine that we prepare doesn't work, right? When we are getting ready to make our flu season's vaccine, we're going to look at what's happening in the southern hemisphere, say in Australia, and see what major strains are infecting them. Some years this is going to be wonderful and work well here in the US, but sometimes there's enough antigenic shift between what they saw in the southern hemisphere and what we see in the northern hemisphere that the flu virus doesn't work all that well. Okay, and all of that is because of the way that the segmented genome can switch pieces and make new strains. Kind of cool. Okay, so let's talk about the flu virus's replication cycle. Now, one of the things that it should be noted about influenza is that it is an RNA virus, meaning that it has an RNA genome. So when it goes through this replication process, remember, we do not have a RNA polymerase that will proofread it, so we're going to get more and more mutations because it's an RNA virus. And that's also going to lead to how many strains we have every year, because it does mutate very quickly on its own that way. Now, influenza has a very specific tissue tropism. It only infects upper respiratory cells, and there's a reason for that. Okay, and that reason is this HA spike that is on the outside of the virion. Now, we named the flu virus after these different HA spikes. Like, if it's H1N1, our HA spike is the H1 spike. And the N1 is going to refer to what we call neuraminidase, and it's going to be what helps the virus bud from the host cell. Okay, so if it's N1, it's the N1 neuraminidase. Okay, so we have this HA spike that is going to bind to the receptor proteins, and the virus is going to come in via endocytosis, and the endosome is going to fuse with the lysosome, and what's going to happen is the DNA is going to be released into the cytoplasm. So we have this negative stranded RNA. It's going to be released with the RNA-dependent RNA polymerase, which are then going to move into the nucleus. Okay, so some of it is going to be red, and we're going to make proteins of that. Okay, some of the components are going to go into the nucleus and help with making new proteins, but some of them are going to move on to the endoplasmic reticulum and the Golgi apparatus. These are going to be the ones that are specific for that HA spike. Okay, and they're going to bud off from the ER and go to the Golgi apparatus, and then they're going to go from the Golgi apparatus, and they're going to make a home for themselves in the cell membrane. And we'll come back to what this does with the virus here in a second. Okay, so we have the mRNA that gets transcribed and translated, and then we're going to get the mRNA that gets replicated. Okay, so it's going to go from negative sense to positive sense, and then it's going to be made back into negative sense, which then can be bundled up into the new virions. Okay, so we're going to have some of these proteins that were made come back into the nucleus. They're going to help package the RNA. Okay, and then they're all going to move to the cytoplasm and move towards the plasma membrane. So those HA proteins that were made and went through the ER and the Golgi apparatus that are now in the cell membrane, these are going to have some of these packaging proteins line up on them, and this is what's going to make the new virions. So what's going to happen is that N1, that neuraminidase, is going to cause this virus to bud off, and it's going to make an envelope of the host cell's membrane. This is why the flu virus can destroy upper respiratory cells really quickly. okay, because it's literally taking pieces of it as it goes. Okay, so this is how we get that enveloped protein with this HA spike in it. Okay, so what's interesting is this neuraminidase is very specific to the flu virus. So we have some treatments that are going to target it. The biggest one is Tamiflu. So it's going to target this neuraminidase, and it's going to prevent the virions from exiting the cell, and the immune system is going to come and destroy that infected cell before it can release its new virions. Okay, so that is the cycle of infection for the influenza virus. Now, one of the things about the influenza virus is it's very infectious, so you can catch it really quickly, it spreads very quickly, but our immune response is pretty fast too. Okay, so a healthy immune system can clear a flu virus infection within 1 or 2 weeks. More often than not, our immune system is going to be a little bit primed, even if it's a new strain, because it evolved from past strains and has some similarities. But even then, our it's not virulent enough that it's going to cause us a lot of damage before our immune system can kick in. This is not like HPV, which can hide dormant in the cells, and that has a long-term cycle where you never really get rid of it. So the flu infection will go away within a matter of weeks and likely not come back. You don't really get infected with the same strain twice. Part of this is because of your immune response, and the second is because of that antigenic shift and that high mutation rate. Okay, so this is the flu virus. That ends chapter 12.5 for us. We'll see you in 12.6.